A convergence of technologies is happening now that holds immense potential for helping human health. These are artificial intelligence, synthetic biology, robotics, and miniaturization. There are approximately 100 million scientific papers ever published, and of those, 30 million papers are in biology. We propose to use artificial intelligence with retrieval augmented generation to solve various biological problems. There are many such problems in biology that we could attack, such as life extension, development of vaccines, treatment for rare diseases, development of new antibiotics, and cancer. There are many different modalities of cancer treatment. One new platform that has been developed recently are BiTEs, or bispecific T-cell engagers. Tebentafusp P is one of the first BiTEs to be approved by the FDA for treatment of uveal melanoma, a particularly aggressive eye cancer. Others have been approved and more are in development. The important part of this is the GP100 protein, an antigen that is found on uveal melanoma cells. We researched this and found that papers published long before Tebentafusp P was developed had mentions of the GP100 protein. We propose to do this on an industrial scale and try to cure all cancers, starting with the most low-hanging fruit. We propose to work with partners such as Ginkgo Bioworks and Twist Biosciences, who are working on automating and miniaturizing synthetic biology. The chip technology developed by Twist allows for millions of reactions to take place on a single chip, something that would take a large amount of laboratory space using traditional equipment. For this project, we have developed a web-based artificial intelligence agent that will do the following. 1. Search PubMed Central for full-text papers about a chosen cancer. 2. Process these papers using artificial intelligence and identify all potential antigens in these papers. 3. Use artificial intelligence to develop a robotic laboratory procedure using the Ginkgo Bioworks platform to generate antibodies using phage display that can bind to these antigens on MHC molecules. 4. Generate patents with placeholders in them for the sequences that will be generated on the Ginkgo Bioworks platform. 5. Write a business plan for a company that is going to be using a BiTE therapy to cure a specific cancer. For our demo, we first input the name of the cancer we are interested in studying. For this example, we will use IDH wild-type glioblastoma, an often aggressive form of brain cancer. After we type the name of the cancer, the AI gives us a summary of the cancer and does an analysis of how appropriate BiTE therapy could be for this cancer. After this, the AI does a search for any PubMed Central papers that would be helpful. This process can take a considerable amount of time. Luckily, in this case, we have results already cached in the system. After this, the AI then searches through these papers to find any antigens that appear to be overexpressed in this cancer. Here again, this step takes a bit of time to search all the 581 papers, but we can use a cached version. The AI then develops pseudocode for the Ginkgo Bioworks platform to carry out the experiments necessary to create new antibody candidates and to sequence them. This process would take weeks to complete. The output of this would be a number of sequences of antibodies. We then write patents with placeholders for the sequences for the antibodies we create. We then write a business plan for the business.